We are looking at what we see as being the most important biological obstacles there are to treat brain tumours more effectively. We believe it's very important that we have an all-human laboratory. When I compare this with some of the old models, they're poles apart in many respects.
So we want an all-human model. So it is quite important for us to know how the human cells work to make up the human model. Because different species work differently or will behave differently. Everything that we use is a human-derived component. So the cells themselves, those are grown in specialized media, supplemented with human serum.
We work on something called the blood-brain barrier. Blood-brain barrier is very important because it protects the brain from toxins, and that's protected by the way that the blood vessels within the brain are different in their form compared with blood vessels in the rest of the body. So they stop these nasty substances getting in. We're reconstituting a model of the blood-brain barrier which we can keep alive over a number of days using human cells, and we can use that to study the passage of drugs across that barrier. So we can transiently open that barrier to let drugs in, or we can use tiny particles called nanoparticles to deliver therapies which wouldn't otherwise get into the brain. And then we can also use it to look at cancer metastasis. So breast cancers and lung cancers, and even kidney cancers, can and do spread into the brain. And when they spread into the brain, the prognosis for patients takes a dive. So if we can stop them from spreading to the brain, we can give those patients a better expectation of longer survival or cures. So how we're doing that is we're growing these blood vessels artificially, then we're squirting cancer cells through them, and we're seeing how the cancer cells adhere to the blood vessels and then force their way through. And we can monitor that using special physiological pieces of apparatus over several days. So I think we believe it's very important that we have an all-human laboratory. And obtaining clinical tissues is critical to this whole process. We have an amazingly generous ethics permission, so we can do many different studies. That's jointly held with King's College Hospital in London, where I used to work. So what happens is we send them up boxes which contain a tube for peripheral blood sample and another tube with a holding medium, which is used for them to take out under sterile conditions during the surgical procedure on the patient brain tumor tissue and put that into this little tube. Then this is sealed, it goes in the post. 24 hours later it turns up on our doorstep. We take the blood, we process the blood, and we grow those cells in the laboratory. It's really important that we maintain this research. I think this will give us results which are more reproducible in the human. And one of the things I really truly believe in is that we can move these repurposed and reformulated drugs into clinic without recourse to use animal models. Because at the moment, we're always told, oh, you need to validate this in an animal model before it'll go into patients. Well, I think that actually, particularly the repurposed drugs which have already been used in patients, we should be able to get them through a very highly sophisticated three-dimensional model of the brain without using those animals.
To all the supporters and people who donate to Animal for Research, thank you so much for donating money to make this research possible.